So our next speaker is Craig McDonald. Craig McDonald comes from California, from UC Davis, where he was one of the leaders, the PIs of Sarepta's gene therapy study. So Craig?
So today we're going to discuss the recent FDA advisory committee on gene therapy. It's really, I think, an exciting development, an exciting story. I'm going to give you a bit of an inside perspective as to what has transpired over the last few months. And really what we're going to focus on today is really the power of advocacy and real-world evidence that can really, I think, provide important perspective and inform an FDA advisory committee on gene therapy. In this case, for Delin-Distrogene, moxicarbovec, elevitis, I believe.
Yeah.
And so this was actually the first gene therapy advisory committee that was ever held in the Duchenne spectrum. It was held on May 12th by CBER, which this is specifically the Cellular Tissue Gene Therapies Advisory Committee in response to Sarepta's BLA. And just to give you some events leading up to the virtual AdCom, Sarepta was initially informed by the FDA that an AdCom would not be necessary, and that was a little bit surprising. Perhaps at the time, some of the reviewers of the FDA perhaps didn't have the most positive view of the data. And subsequent to that, a group of academic physicians, myself, Craig Zaidman, Crystal Proud, Perry Shea, Jerry Mendel, who had treated the most patients with the Sarepta gene therapy product at the time, provided a letter of our clinical observations, what we were seeing in the patients. And also we provided some pre-treatment and post-treatment videos on a Vimeo site, and we directly sent this to Peter Marks and Celia Witten, and they provided it actually to the FDA review team. I'm not sure if that had any type of impact. 2 weeks after Sarepta was initially informed that an AdCom would not be required, the FDA reversed. There we go. The FDA had reversed their decision and decided that they would go ahead and hold an AdCom for the purposes really of transparency and provision of an open discussion of an AdCom, since this was really the first BLA that had ever been put forth for gene therapy in Duchenne muscular dystrophy. And then subsequently, 4 of these academic physicians, we were actually able to meet with Peter Marks at the MDA meeting in March of 2023 and really share our experiences with him for about an hour one-on-one. He really listened. He also assured us, at that time we knew there was going to be an AdCom, he assured us that the discussion and voting questions would be fair and balanced, because we had some concerns about that, but just based on the previous AdComs that had been held in the Duchenne space. And just a few observations that I'll make is I've been an academic physician, expert in all 4 of the AdCom meetings on Duchenne therapeutics. The final FDA discussion questions and voting questions were really excellent. I think they were very fair, they were very balanced. And I think another really important point is the AdCom chair, Tabi Asan, who's a biomedical engineer from City of Hope, I think really was an extremely effective and even-handed chair of this AdCom meeting. The Sarepta team and experts was really a cast of over 100 individuals, went into this AdCom extremely well prepared, and I think performed in really a spectacular fashion during the AdCom. But I will say that just by the way the processes are set up for these AdComs, the FDA essentially has a bit of a home court advantage. They can continue to experience tactical advantages in the AdCom process. Just to give you an example, the sponsor has to provide the FDA with their slides that they're going to present the AdCom 1 week before the AdCom meeting. The FDA provides their slides about 30 minutes before the AdCom meeting, just as an example. The sponsors, I think the DMD advocacy community and patients, I think, really won the day based on compelling data, real-world evidence, and inspiring experiences that were put forth in the open public testimony. Sarepta elicited input from James Signorovich and Chris Mullen, really talented biostatisticians who provided invaluable input. And then Christopher Buddy Cassidy, a Duchenne patient, as a temporary voting member of the committee, was really an effective and heroic advocate for the Duchenne community. You are all enormously should be proud of him. You're going to hear a lot from Buddy today, and I think you're going to hear also Buddy's going to be speaking on Saturday, which I think will be really exciting. And the Duchenne advocacy community, I think, provided really compelling public testimony and really very compelling videos, which I think were noted by the advisory committee members. And I'd like to just say that I think Peter Marks and likely Celia Witten, the director and the deputy director of FDA CBER, I think really made courageous decisions for Duchenne patients during the process. The voting question was fairly straightforward. Do the overall considerations of benefit and risk, taking into account the existing uncertainties, support accelerated approval of SRP-9001 using as a surrogate endpoint expression of Sarepta's microdystrophin at week 12 after administration of SRP-9001 for the treatment of ambulatory patients with DMD and a confirmed mutation in the DMD gene. And just to summarize, I think the FDA's position, there were some manufacturing issues, process A versus process B, which ended up really being not significant after all. But in terms of the surrogacy of the microdystrophin expression, I think that was really a major issue for the FDA. They were concerned about biological plausibility. These aren't naturally occurring dystrophin-like proteins. They lacked some potential important functional domains. There wasn't necessarily the animal empirical evidence that was robust. And in terms of the clinical studies, they felt it was challenging to distinguish the effect of SRP-9001 versus standard of care. And it was really unclear which patients might benefit from the gene therapy, and they felt there was no clear association of microdystrophin and change in NSAA score. As it turns out, safety was really not a significant issue, I think, for the FDA review team. During the AdCom, they did bring up the concern about potential cross-reactivity to future gene therapies, making patients potentially ineligible, and also brought up some AAV-based gene therapy, some safety concerns just based on AAV-based gene therapy products as a class, but not specific to the Sarepta compound. And then there was this issue brought up about the ability to complete really the EMBARK confirmatory trial if they provided commercial access as of early June. And then they finally stated the uncertainties make it difficult to consider Sarepta's microdystrophin as a surrogate endpoint reasonably likely to predict clinical benefit in support of accelerated approval. Data from phase 3 will be available later this year and should help clarify these issues. Patients likely will have only one chance to receive an AAV vector-based gene therapy. It's critical that it is effective and safe. So I think in all, the FDA was certainly rather negative going into the FDA AdCom. And here is the Sarepta team. As you can see, they are primed. This is about 30 minutes before the AdCom. They're well prepared. They're excited to state the case on the behalf of Duchenne patients. And really the first discussion question really focused on this issue of surrogacy of the microdystrophin and the use of the microdystrophin on biopsy as an endpoint reasonably likely to predict clinical benefit in ambulatory patients with DMD. And I think as a conceptual framework or construct, we had come up with this prior to the AdCom, which I think was really useful in terms of providing realistic expectations for patient outcomes. This is the North Star shown on the y-axis by over time. The maximum score is a score of 34. And patients that are in the skill gaining or maturational phase of the disease A treatment benefit would actually be seen by showing incremental increases in North Star over that seen in the untreated placebo patients or the natural history patients. Whereas if you have patients that are in the functional declining phase of the disease and have already lost significant muscle fibers, I think our treatment expectations we've learned with gene therapy tend to be different. What we see is stabilization of functional decline in a patient who's really expected to have a pretty consequential and pretty significant functional decline. So stabilization of disease, and then followed by long-term durable effect or long-term stabilization, hopefully for many years. We don't know how long, as Anamika noted. So if you look at short-term treatment benefits, the short-term treatment benefits can actually be fairly comparable if you look at an appropriate control population over the short term. But as you follow patients longer, and the only way you can follow patients longer is with an open label follow-up, not a placebo-controlled trial, but as you follow patients longer, the treatment benefit actually becomes greater over time. And you heard the FDA talking a lot about just moderate treatment effects, but as you follow these patients longer and they actually demonstrate disease stability, the treatment benefit actually becomes quite dramatic over time. And then when you start looking at things like correlation between change in North Star and the amount of microdystrophin protein, just think about it mathematically. If you look at the change in North Star in this patient population that's in the functional improving stage of the disease, they're going to actually show, the FDA was really just looking at just absolute changes in North Star. They weren't really looking at changes in North Star versus natural history, historical controls, and so forth. So these patients would actually have an increase in their North Star score. These patients actually have a change in North Star of 0. So both these patients are benefiting. They're both expressing dystrophin, but just mathematically, how are you going to get a robust correlation coefficient when one patient is showing a 0 change but still benefiting, and the other patient is showing a positive change? And I really think that's an issue that the FDA had missed on. This was the FDA slide where they actually showed they felt there was no clear association between Sarepta's microdystrophin expression and change in North Star score. However, the 4 to 5-year-olds, if you look, they've got robust microdystrophin expression and also very significant changes in their NSAA score shown here. If you look at the greater than 6-year-old population, those patients are expected to be stable. There's really no change in their North Star score. They were showing changes in their microdystrophin values. You see there the FDA is really focusing on just a change in total North Star. They did state, however, that limited data suggests an improved change in North Star total score with increased microdystrophin expression in younger patients 4 to 5 years of age. And that's ultimately very similar to their approved label. And again, here's a slide from the FDA, really concerned about the open label design without concurrent placebo control. Again, they're focusing on just a change in that total North Star score, and they conclude that the correlation is not sufficiently persuasive to consider expression of Sarepta's microdystrophin reasonably likely to predict clinical benefit. And Sarepta put forth, this is their correlation data. As expected, it's not a strong correlation. Again, because of some patients are actually benefiting by having 0 changes in North Star. Some patients are benefiting by having a positive change in their North Star. But I think in general, the microdystrophin expression was really quite robust, and there was a statistically significant relationship and also a saturable response. So as you get upwards of 40 to 50% or higher, you don't get an incremental change in the North Star score because it's a saturated response. You kind of reach a plateau, and that's going to affect your correlation coefficient as well. So question 2 really focused on the randomized placebo-controlled data, and the FDA here really noted that the ambulatory patients, there was some signal of treatment effect. However, among ambulatory patients 6 to 7 years of age, there appeared to be no difference between the SRP group and the placebo group, and no improvement from baseline. And they also mentioned that the NSAA score is effort dependent and process dependent. So they note that the results of open label studies are difficult to interpret because of this effort dependent nature of the NSAA. So they felt it was affected by motivation and effort. And really the literature would not support that at all. There's a lot of literature that shows that natural history data on the North Star is very similar to the placebo-controlled data coming out of long-term clinical trials in Duchenne. They also thought that there was perhaps process-dependent issues, but we know that these tests are rigorously obtained. There's rigorous training that goes on by Adam and other clinical evaluator trainers. They also mentioned, the FDA, that microdystrophic expression wasn't really analogous to any clinical benefit. Again, they support that the open label single-arm studies were difficult to interpret. Clinical endpoint were effort dependent, process dependent, and so forth. And here they really even just cited their own guidance in terms of the need to conduct placebo-controlled studies with blinding because of this issue of coaching and encouragement by family members, caregivers, and medical staff, who actually in these trials were not really allowed to be in the evaluator areas when these patients were being assessed. And so I think Buddy Cassidy really, I think, focused really quite eloquently on the validity of the NSAA. We didn't really have the opportunity to respond because our Q&A response time had actually passed, but Buddy was really great in this regard, commenting on the North Star. If we could just turn up the volume a bit there.
Cassidy?
Yes, so I'm still not convinced by the FDA's explanation as to why they question or are skeptical about the validity in certain areas of the North Star ambulatory assessment. Certainly, open label conditions would have an impact on any kind of performance evaluation measure. So that would be like that stands for performance evaluation in general, not the NSAA in particular. And so, I mean, you can't really get, you can't make it perfect, but you can only mitigate influence of suggestive bias. And the literature shows, it's well documented, for the NSAA does a remarkably good job in creating protocols to guard against bias. And again, this is well established in the literature. So I mean, the problem of open label conditions influencing things would be problematic across the board for this mode of evaluation. But we need to look at the NSAA in particular. And in fact, in the NSAA evaluation, this would be less of a concern than it would be with other evaluation criteria. And again, this is well documented by a number of different scholars, Riccati, Henriksen, Duong, and these names are in the sponsor data. And I'm just seeing industry guidelines in the FDA and investigative report. So I would say that certainly open label conditions are a concern, but the NSAA is constructed to guard well against subjective bias.
Great, thank you.
So really pretty impressive explanation and knowledge of the literature, and you know, we could not have made that point better, and so that was huge. Also, just note, this is the FDA slides. They discuss that SRP-9001 did not appear to have an effect on 6 to 7-year-old boys with DMD. And just look at the area highlighted there. They say, but SRP-9001 group showed no improvement from baseline. Well, if you remember, stability is a win here with these patients that are in the decline phase of the disease. So I think that was an issue with regard to their interpretation. So Duchenne disease progression is heterogeneous, but it is predictable. There's this consistent pattern over time where patients are actually in a functionally improving stage. They peak and then they functionally decline over time. And actually, we can predict their trajectories by using baseline characteristics, things like 10-meter walk-run times and time to rise from the floor. And just to remind you, again, our treatment expectations are different if patients are in the improving or functionally increasing phase of the disease versus the functional decline phase of the disease. And just to give you an example, this is our natural history data from CTAP. And if you look at time to rise from the floor, this is the trajectories for patients with different disease severity, the red being the most severe, the green being the least severe trajectories. And what you see is once you get above 5 seconds, patients decline and really quite rapidly. But when you're below 5 seconds, they actually are either improving, quite stable, or just showing more modest decline. And in fact, when you look at the randomized placebo-controlled 102 trial that was conducted of the gene therapy, the 4 to 5-year-old groups was really well balanced. There was a statistically significant effect of SRP 9001, a 2.5 point change on the NSAA, but there was this huge imbalance in the placebo group versus the gene therapy treated group. And both actually remained stable, but the placebo group were much higher functioning at baseline. And so you can see the North Star scores were different. And in fact, if you look at their time to rise scores, the placebo patients were 3.6 seconds. The treated patients with gene therapy had a baseline rise floor of 5.1 seconds. And we know based on the natural history data on the right that the patients with the 5.1 rise from floor time, they're the blue trajectory in terms of loss of ambulation, whereas the other patients are in the purple trajectory. So they have very different trajectories. In fact, the Sarepta gene therapy treated patients were really destined to deteriorate, whereas the placebo patients were really predicted to be stable over the course of the year trial. And this just outlines the profound imbalance. The yellow dots are the placebo. The purple dots here are the gene therapy treated patients, and that red line there is the 5-second line. And you can see that there's not a single placebo patient that was above the 5-second line, whereas a number of the gene therapy treated patients were well above that 5-second threshold and were predicted to functionally deteriorate. A statistician during one of our preparation sessions actually quickly did some statistics on this. Bad luck for Sarepta and bad luck for the Duchenne community. He said that the probability of this imbalance occurring was about 1 in 1,000. It's just really unfortunate that there was this degree of imbalance in baseline factors. But yet the FDA said, the best way we could really make the case was comparison to external control data. And they mentioned this issue of effort-driven, process-driven characteristics of the NSAA. And so Sarepta brought forth external control analyses. These were pre-specified prior to unblinding of data. There is a risk of potential bias, but the FDA had really a different interpretation, did not really view this as pre-specified. They viewed it as not statistically rigorous, not pre-specified for hypothesis testing, did not use a pre-specified multiplicity adjustment strategy. It was a lot of statistical rationale in terms of their take on this not being rigorous. But Sarepta did actually use a very rigorous external control selection. They took patients from 3 clinical trials, the Synergy Natural History Study, the Lilly Tadalafil study, and the 4-DMD steroid trial. They used similar inclusion criteria and so forth. And there was a lot of patients actually that provided external control data. And so if you look at these patients in the commercial grade Sarepta open label trial, study 103, they actually have a 3.2 point gain relative to the external control. So really a quite dramatic gain. And so again, when we look at the study 102 data, taking this into account, the 4 to 5-year-olds versus the placebo group actually show that incremental benefit, statistically significant. Interestingly, when you look at the 6 to 7-year-olds, the placebo patients actually perform very similar to their external control cohort that had baseline prognostic factors. Whereas look at the Sarepta gene therapy treated patients, they actually remain stable versus the external control population, which is actually exactly what we would predict in a patient population in the declining phase of the disease. And both treatment deltas were very similar and significant p-values. And then the long-term data out of Nationwide Children's on the 4 patients followed for 4 years now, periods of about 5 years, show improvement in function and then stability long-term, which is really quite impressive. And actually, when you start to look at the treatment magnitude here, a 9.4 point change on the North Star long-term when you follow these patients out for 4 years. That is a substantial magnitude of a treatment effect in these patients. And just to show you some illustrative videos, these were shown to the advisory committee meetings. This is a patient pre-treatment. This patient's 2 days short of 6 years of age. Here he is struggling down the stairs. Here he is 6 months post gene therapy treatment, doing stair climbing. You can see the ease at which he's walking down the stairs. And then on the right, you can see him actually climbing up playground equipment really quite easily. You literally couldn't pick this kid out from other kids on the playground. Just the ease at which he's going down stairs and then running in the playground environment. And Dad actually is a tech-savvy person, got himself a drone with a camera and actually videotaped this kid, running on the field and just sustained running with really quite well-maintained endurance. Really pretty impressive. Here he is here he is actually riding a bicycle as a newly acquired skill. Here, 2 years, same child, 2 years post-gene therapy. We didn't show these videos in the AdCom, but here he is in terms of his rise from the floor, 2 years post-gene therapy. And here he is, hopping. And, again, Buddy comments on the videos and the importance of disease stabilization here.
In terms of FDA's guidelines to industry for Duchenne, it does mention potentially looking at patient-reported outcomes to assess clinical meaningfulness. I guess what I'm concerned about here is that in terms of the investigative report, we're talking about accelerated approval process for a relative disease. And it doesn't seem to me like there's much of a place or room for a discussion of qualitative changes outside of the clinical setting. And I'd like to stress, as the patient representative, that these things are so important and meaningful to patients. And I would just like to stress that qualitative observation is indeed scientific, and it is not to be conflated with anecdotal evidence. Again, I think quality of life is very important here, and we need to seriously take into account what we're seeing in the videos. In terms of the Gower sign, there are actually documented instances of patients no longer using the Gower maneuver to get up. And I can speak from personal experience that having had to use the Gower maneuver myself, which would put me at about a 1 on that NSAA evaluation, to a 0 not being able to get up on my own at all. And that's not just significant, but almost miraculous that we as Duchenne patients, in terms of the natural history, in terms of lived experience, don't get better. We don't improve, right? So to not do that anymore is, to me, nothing short of miraculous. And
So, I think really pretty eloquently spoken there for sure. And, just in terms of, we have other examples of patients treated with this therapeutic who are, running and really quite functional, many examples here. And even patients older who are actually showing stabilization of running ability, not normal running ability, but stabilization after 2 years of treatment. And so I'd like to just conclude here. I think Buddy really made the point that time is muscle. Stability is really a critical feature here. The FDA Advisory Committee ended up recommending for accelerated approval by a vote of 8 to 6. They did approve it for 4 to 5-year-olds. And I would just refer you to Peter Marks as a recent memo really providing his rationale for that approval and their rationale for not approving it for the older children. And just to conclude, I think these advocacy perspectives, real-world evidence, were really critical to informing an FDA advisory committee and FDA leadership on the risk-benefit of gene therapy. The approval of the first gene therapy product for the treatment of Duchenne was historic. Time is muscle. Stabilization is a huge benefit for patients with Duchenne. And while the approval is limited at the moment to 4 to 5-year-olds, it's certainly disappointing. It doesn't appear to have biologic rationale. It is a huge first step and a huge win. And with more data forthcoming towards the end of the year, hopefully we'll be able to provide this therapy to other patients, and hopefully this will provide a pathway to other sponsors bringing forth gene therapy treatment. And with that, I'll conclude. You'll hear more from Buddy on Saturday, but thank you very much.
Thanks, Craig. So get your questions ready, because we have Annemiek and Craig and Kevin to answer any questions you have before Chris Jones will give his keynote. But I have a commercial to tell you, or an announcement. So people who are trying to access the Wi-Fi need to use the conference Wi-Fi. The password is all caps, PPMD23, because you can't access the hub from your room Wi-Fi. So it has to be conference Wi-Fi. The passcode is PPMD, all caps, 23, and then you could access the hub. And with that, who has questions?
I have a question for Kevin. We've been hearing a lot about the infant that you treated and a number of people we've talked to, and now that that infant is 19 months old, I'm wondering if he's walking.
Sorry.
Are you on?
Yeah, he is. He's normally he's doing completely normal development. He's not, we can't tell him by Bailey or other scores from a normal infant. Yeah.
Other questions?
Hi, question is also for Kevin. You'd mentioned that the CRISPR technology is able to make the cut and sew back basically the dystrophin gene. And you mentioned that the PACE part, I guess putting back portion of the gene, is that something that any research has been done on that part, or?
Yeah, there's an enormous amount of research using CRISPR-based technologies. And I apologize, I went through it fairly briefly because it's actually fairly complex. There's ways to design CRISPR-based therapies to cleave out parts of the gene, to correct individual spots of the gene, and potentially to put fragments back in. I was trying to say that all of these are being explored by different groups, and there's newer technologies called prime editing and base editing that people are exploring. It's kind of that there's not one overall solution to put the whole gene back in place that exists right now. But there are many approaches that are likely to be tried beneficially for different little groups of mutations, but each of them faces kind of the same questions about safety and immune responses to these foreign proteins that we're putting in, and There's not just one CRISPR approach. That makes sense.
Yes, thank you.
So I have a question, if you don't mind. And I know, Annemiek, you're involved in some of this, and I know you've heard of this, Kevin and Craig. So can we, should we, or when could we have a designer therapy? So because as you know, there's multiple mutations here. We're hearing about N=1 trials, N=few. How do we get that done? How long does it take? And what's possible?
I can start on this. So I think, so if we want N=1 or N=very few, it's really important that we first have convincing data, also functionally, for the approaches that we have now. So if we're talking about exon skipping, the problem now is that the dystrophin levels that we restore are very, very low. And I think the levels that Kevin showed with the duplications, those are levels where we think, okay, if we see 8%, then that has to do some functional things, and that's also what Kevin showed. So once we're there for the other exon skippers, then of course it will become much easier to convince the regulators. If we show for 2 patients, look, we do this, and then we see 5% dystrophin or 8% dystrophin, and look, we showed in all these other trials, if you have that amount of dystrophin, then you will get a functional effect. It will be much more easy to convince them than now where we say, well, we see 1% dystrophin, well, will that slow down the disease progression? Yeah, we think so, but we don't really know. We don't know to what extent. So I think really this will rely on having more data, and then we can build on that. And if you look at other N=1 examples, they're all building on, for instance, nusinersen for spinal muscular atrophy, where there is convincing data. Patients live longer, and that's, of course, that's hard to argue against. And then based on that, you can say, well, maybe we can tailor it for other diseases as well. So I think it's really important to bear in mind that if we want to do this for single individuals, we first have to show for a larger group that the approach works. And what amount of dystrophin do you need? If you know for the microdystrophin that you need 35%, for instance, that is the critical threshold, that will also have implications for other muscle diseases that miss dystrophin binding partners, for instance.
Thank you.
So I just want to add to that. I think one of the challenges when we think about personalized therapies is the models now for approval to deliver these are based, right now when we develop a new therapy, we go through cell-based therapies to pick our candidate, we go through animal models to show safety and efficacy. And how do we make a jump? It is not feasible in the long term to make an animal model for every single person's mutation. We're engaging with the FDA soon on this question. The U7 approach I mentioned is particularly amenable to a rare classes of mutations where, for example, there's mutations deep within an intron and a piece of the intron is included as a fake exon within the gene. That group of mutations is a perfect candidate to do individualized therapies because if you can't do any wrong skipping in that region, and you would make wild type dystrophin. But it's not feasible to make a mouse for each of those. So we're engaging with the agency to try to get an answer. How can we build on our success with Duke2? How can we use that data to move from human cells, skin-based cells where we do the testing? How can we make that jump into patients in a safe and compliant way? And there are people out there who says, you give me a couple $1 million, I'll make a therapy for your child. And I think that's not necessarily, there's a lot of cautions in this process that we have to really be aware of. I think there's potential to do it, probably not for every rare mutation, but for a lot of rare mutations, we can do that. And we're exploring that pathway with the FDA to say, what's the regulatory path for it now?
So you'll know more soon, right?
Yeah, I anticipate by this time next year, we could give you a more clear idea of examples where the feasibility of that case, yeah.
Okay. And Craig,
Kevin, if you were to pursue an accelerated approval pathway, would there be a requirement for a confirmatory study of some sort? Or would it just be durability of effect? Or
for this one, for duplication 2, yeah. Well, it's a little complicated. So we are a little hamstrung by the fact that we only had enough virus to treat these 3, right? This therapy I'll mention was licensed to Astellas, who stopped all their Duchenne programs, and we may have a new licensing partner for it to move forward. There is a pathway. I think, you know, I'm speaking out of turn here to say I want to be clear. We're not doing this tomorrow. This is not there. But one could imagine how you could treat another half dozen infants like this, show this degree of restoration, show entirely normal development, and you probably would be able to move forward with an accelerated approval based on those individuals. Full-length dystrophin as a biomarker is different than microdystrophin as a biomarker. So, but programmatically, we still would want to explain why is it that older boys did not respond the same way. We can think about why that is. There's more fibrosis in muscle and so forth. And we have safety range, I'll say, in our preclinical studies to be able to dose higher in those boys. So it's a great question. It's really a regulatory question, and one we have to discuss with this future licensing partner about.
Okay, more to come. I think there's a question here.
Yes, hello. Thank you for all of this information. It's fantastic. I'm a mom of a 16-year-old with Duchenne, and have to say that we chose, when he was diagnosed, to administer steroids, initially based on the anecdotal evidence and then based on some of the study information. But it's been really hard to watch the side effects of the steroids. And I'm wondering if you can comment on how some of these newer therapies are going to impact the need for steroids.
Well, my opinion, sorry, you go ahead.
No, I wasn't going to comment.
My opinion is we don't know yet. That's in brief my opinion. I don't know what your...
Yeah, I think certainly there are emerging therapies that target the inflammatory cascade, that are immunomodulatory, and have potentially better side effect profiles. I think that there's some exciting work emerging on perhaps even different steroid regimens that might mitigate some of the toxicities. Even people are looking at even once a week steroid dosing. I think the next really interesting, I think, set of emerging questions is going to be, if we have a population of patients that have been treated with gene therapy and actually have relative stability of function, could there be clinical trials designed, random withdrawal type trials where you actually ask the question, is gene therapy potentially steroid sparing? And I think this is going to have to be looked at, in a somewhat safe and systematic manner. But I think it's not, unreasonable that perhaps, we could at least mitigate the toxicity by perhaps, getting into some different regimens in the setting of some of these precision medicine therapeutics. So I think that'll be another set of really interesting questions. But I think there are a number of new therapeutics on the horizon, which I think may allow us to decrease the toxicity that we're seeing with steroid treatment.
I agree entirely, and I want to say the one caveat I think is that the one way one loses the effect of gene therapies, transgene therapies, is when that fiber degrades, that episome, that vector genome disappears. So we have to be cautious in how we think about these trials. Withdrawing steroids and allowing inflammation to occur in what now is something like a Becker-like muscle might actually promote, in a way, loss of vector genomes. It's actually a complicated issue, and we'll have to be really thoughtful about how we design trials like that to answer it.
And even some of the selective fast myosin inhibitors that companies like Edgewise are actually developing for both Becker muscular dystrophy and Duchenne muscular dystrophy could theoretically have beneficial effect in patients that have had dystrophin restoration strategies and treatment.
Yeah.
Question over here.
With just so many therapies, coming, we have 6 approvals now. A boy like my son, who is 4, he did start exon skipping when he was 2.5. And my question is, with him being in the group of the 4- and 5-year-olds, are they going to be able to be used together? Will there be a washout phase, and then he'll be able to add the exon skipping after? I don't know if that's been discussed or talked about, but that's been on my mind.
Did you get the question?
That's a tough question, and I feel for any parent in that situation. I want to add that depending what specific exon skipper your son's on, there are trials starting right now of sort of generation 2 of exon skipping drugs that promise from the preclinical data supports the idea that they're much more efficient than the current version 1. So Dine's trial or Validity's trial, for example, for 4451, the preclinical data very promising for these. And you know, particularly if the levels, if those drugs are as functional as the preclinical data suggests, those particular versions of dystrophins that would be expressed, let's say a 49 to 51 deletion, if you have 49 to 50 and skip 51, that's a very, very functional version of the protein, and one that we can reasonably expect to be more functional than the microdystrophin protein, based on what we understand of observations in humans who have that mutation. So it's a complicated issue, because your other option is washing out and enrolling in one of those trials as well, right? So, you know, obviously I think any of the trial usages of any of these antisense, of any of the gene therapies will require washout. It's a different and more personal decision now that there's an approved therapy, what you might choose to do. And I would advise you to talk carefully to your clinician or clinicians who spend a lot of time thinking about the genotype, the mutation, and the function at the outside in making that decision. Craig, would you, I'm curious if you,
Yeah, I would absolutely agree with that.
But do you see combinations? Like you would start an antisense, get some dystrophin expressed, and then do a gene therapy, or do a gene therapy and then an antisense product?
Well, you know, there's no mechanistic reason once you've got microdystrophin, let's say, expressing. There's not a mechanistic reason that you would lose the function of that by putting in the antisense. It acts at a...
SMA patients, some of them have had gene therapy and are also on the nusinersen, so they have both the gene therapy and the antisense therapy.
So are they complementary, would you expect?
And they're combining, and well, that is the question. But I think what Kevin just said, you can also go in one of these trials, then be aware that some of these trials will have a placebo group, so then you will go from the known drug that you're using now to maybe be in the placebo group. So it will be a really difficult decision, and I don't envy you. In Europe, we were facing the same problem, and we've written an article about this, like, what if your child needs exon 51 skipping? What are the different options that are around them that are coming, including also the gene therapy? So if you want, I can point you to that publication, or I can, PPMD can put it online. And we involved patients as well in that paper, because yeah, it's not just you who has this challenge.
I just want to add a coda to that to say, the U7 therapy that I've described, a company presented at ACS GCT, a U7 exon 51 skipping approach that was really quite remarkable results that might be available in a clinical trial context in Q1, just to complicate your decision-making process. Because the other thing that's not addressed in your question is we don't know yet how insurance is going to handle it, right? Your insurance is paying for your exon skipper.
Yeah.
If you switch and get, you know, we don't know how much insurance is going to let you go back and forth. So a really great question. It's one that's a bar afterward question to carry out all the way.
Oh yeah,
sort of
that's a bar question, perhaps. So one last question, and then Chris Jones is going to end this. Did you have a question? Okay. Yeah, run two. But in the meantime, I'm going to ask Craig one, and then you'll be the last question. So Craig, if this is out of school and you can't say it out loud, you're allowed to say that. But I know in the microdystrophin trials, you had open label trials in which there were non-ambulatory patients. Are you allowed to say those patients tolerated it well, they are doing better, or is that proprietary data and I just asked the wrong question?
No, I would say that you know, there's been at least 6 non-ambulatory patients that have been treated. I would say safety-wise, they seem to have at least tolerated the Sarepta product, you know, quite well. We don't really have the efficacy data. I think the jury's still out with regard to that. We do think that we will be able to adequately power non-ambulatory trials based on the performance of upper limb, and you know, perhaps even looking at some other cardiac endpoints may be quite interesting in those populations as well.
Okay, thank you. Last question.
Okay. Hi, I'm Lisa. I'm a parent of a 8-year-old son. He has a nonsense mutation on exon 24. So right now there's none of the therapeutic means able for him. So I have some questions regarding the macrodystrophin. First is I don't understand, as a normal human, like if our muscle break down, if we have some rest, it will recover. I think the researcher just mentioned it's not reversible, but we do saw like improvement after the macrodystrophin. The kids will be able to run faster, and like even the one with like duplication, he will be able to rise from the bed unassisted. So I'm not sure, is that because the definition of reversible, like a cure, is different than improving function? Or does it mean they will never gain their muscle back even after macrodystrophin? Because I'm not in a biological ground, like biology background. So can you clarify that for me, please? Thank you.
I think, so when muscle is lost, it's lost. However, the muscle that is not lost, that can be treated. And if that muscle is very sick, it won't work very well, but then if you restore the dystrophin, that muscle, that muscle might get better. So when you see very young patients improving, that's not just muscle, that's also growth, and I mean they grow taller, so obviously they will walk better, and they mature and they develop. So that is one aspect, and the other aspect is that, well, if you're 18-year-old and you have a few muscle fibers that are in really poor state and can't do something, but you're just in time to, say, prevent that they become fibrotic tissue, then, well, you can save them and you can regain some function. But I think we have to be very careful because, I mean, this young man probably, he'd not lost that function 10 years ago. He'd recently lost it most likely and now regained it. So I think we have to be really careful that we don't think, oh, so when they're in the wheelchair they can go out of the wheelchair. That is not likely. It's likely that things that have recently been lost because the muscle is in this critical stage of just being able to do it to just not being able to do it, that is the part that hopefully can be rescued. But once the muscle is replaced, it can't be undone.
Okay. Yes, so the second part of my question is, after the macrodystrophin, the kids will develop like an antibody to the AAVs. If future treatment available through the same carrier, would there be a way to bypass the antibody and the kids will be able to get another kind of treatment? I'm asking because the company called Tevard is working with Vertex, from what I'm reading online, to develop a tRNA therapeutic for the kids with nonsense mutation. And they're using AAV9 as well. So I was wondering if my son do get the macrodystrophin treatment, will he be eligible maybe in the future for that treatment as well? Or the gene therapy for now is one-time dose. There's no way once they develop the antibody for the AAVs. Thank you.
So we think of it now as a one-time dose, but there is a lot of work going on to understand about the feasibility of redosing and also the feasibility of treating patients who have environmental exposure beforehand. If people get AAV exposure in the community, it's on the order of about 100-fold less than the kind of immunity we generate, 100 to 1,000-fold less than the immunity we generate with giving the vector. But there's an enormous interest right now in ways to do that. So some ways you'll hear coming up are ways that include trials of plasmapheresis to wash this out of the bloodstream. There are small molecules they're peptidases that can be potentially delivered that cleave IgG, at least temporarily, to get rid of IgG so you could redeliver a virus during that time. There are other approaches people are using to think about it to give conditioning therapies like you use for cancers, for example, or transplantation, to try to knock down antibodies temporarily. So all of these are under study. None of them today would we be able to promise to someone that they could definitely get a second treatment. It's highly likely these issues will be resolved in the next 5 years, though. That was the best we think we could say.
Thank you. All right, So we're going to have Chris Jones come up and close this session, and then we're going to go to break. So Chris, thank you all.